Hello, hello, I am Joe the Science Pro, and this is something completely different than I have ever done, probably ever. Um, and this is not necessarily what I would consider um, something that I'm terribly prepared for. This is, like I said, we're in undiscovered country for here, so I am uh, I'm joined by someone that I respect. I consider him a colleague, a friend, Dr. Brian J. Balin. Dr. Balin is the director of the Center of Chronic Diseases of Aging here at the Philadelphia College of Osteopathic Medicine. And I'm going to ask him several questions about his background, his expertise, his research, and really we want to see overall what kind of information perspective we can learn from him, and eventually I will open up the floor to all of you so that you can ask some questions of him and get some answers direct from the horse's mouth, as they say. So I would like to introduce Dr. Brian Balin.
Thank you, Joe. I appreciate the introduction and the opportunity to speak today as well. And hopefully the information I have based on your questions will be helpful for everyone in the audience.
Well, thank you very much, Dr. Balin. I really do appreciate you joining us here. I know that this is definitely a first for me. Have you given interviews like this of this kind before, or is this just like us, completely undiscovered territory for you?
Well, I've actually been interviewed by a number of news stations in the past about our research, which is Alzheimer's disease research, because it is such a very important topic of today. I've also been interviewed by the scientist, and they used a Facebook feed through the scientist, asking me questions about our work specifically and how it actually could potentially help the large percentage of people that now are being diagnosed with Alzheimer's disease, which is about 50 million worldwide.
Wow. So I guess let's begin at the beginning.
Okay.
Why science? Why not engineering? Why not music, art, English? Why science?
It's a really good question. I can only say from my own perspective is that when I was a young kid, about 5 years of age actually, I saw the first movie of my life, and it was the original Journey to the Center of the Earth. And in that movie, they had science people. Now, those weren't biologists really, but they were people interested in biology. And what was happening at the center of the earth, possibly. So it was a bit of a hokey movie when we look back on it now. However, that was the first stimulus for me to actually now entertain going into science, or doing something with science, because I found it intriguing.
So, obviously you do not become a scientist overnight. Was this a decision that was formulated over many, many months, or was this something that you went into one degree program and then transitioned to a different degree program, or were you always laser focused?
Well, what I did was I actually started out being really curious. So there was a lot of different types of science that actually intrigued me. And as I went through the schooling process and through elementary school and high school and on into college, what really started intriguing me is the universe that we can't see, and that is the microbial universe, what all of these infectious agents that we may have around us, and we do have around us, on us, in us, that I was actually intrigued by what they are doing with us and what we are doing with them. And it led me to actually major in microbiology when I was in college. And then I started looking and thinking, well, what would I do after college? So I thought, well, I could go on to med school possibly, or go on to a PhD program, maybe in microbiology. But then I started investigating the brain and thinking, brain functionality, our human brains are so complex that maybe that would be an interest of mine. So I actually then went on to graduate school and received my PhD working on understanding the brain and the blood-brain barrier, which is interesting at that point in time. Once I finished graduate school, though, then it goes on to a postdoctoral period. It's sort of like what a doc would do if they go after medical school, you'd go into residency. So for me, it would be going to a postdoc after my doctorate degree. And that's when I went into Alzheimer's disease research. And I did that because I had an uncle who actually developed Alzheimer's disease when I was fairly young, and he was a favorite uncle of mine. And I said, I think I could make some inroads here of understanding that disease process and hopefully make a difference for the individuals with that disease. And along the way, after my postdoc, I had my first job at the Medical College of Pennsylvania in Philadelphia. And I started my own lab with studying Alzheimer's disease. And then in the mid-1990s, I stumbled into an arena of infection with Alzheimer's disease. And that's what we've been focused on up to this very day.
So you kind of almost came full circle. I mean, I have kind of, I guess, the knowledge of the secret sauce, if you will, because I know what organism you're studying because I'm technically affiliated with what you're studying. I guess it's kind of interesting for you that you majored in microbiology at the very beginning, and you kind of came all the way around to in a way, studying it again. But it's interesting how that journey kind of led you there.
It is, and it's one that you don't really plan out in a specific way. It's through a lot of serendipity, which is how science is still done to this very day. We make observations, and if you follow up on some of those observations, sometimes you make discovery. And with discovery, you can now have a process where you are following a path of understanding. And to me, that's where the curiosity is still there. It's the want to make a difference in the field. It's then to try to understand what is the biology behind many of the different types of diseases we have and that we know about at this point in time.
So I guess one of the next questions that I have for you is I know that some people, when they go into almost a vocation-like situation, they don't necessarily always find the support necessary in order to give them the drive behind them that they need. Did you ever encounter something like that, or were you always supported by your family and friends?
Well, I was always supported by my family. I was always thought to be a little bit different by most of my family, I think, because of how I thought about the world, of science, of medicine, of actually approaching my life in a way of making a difference for others. And that's what I still do. So the support from my family has always been there and been great. Most friends as well have done that. What's interesting is the support that you need from your fellow scientists is where there is a degree of almost an absence. When you basically, as a scientist, go down certain paths that are unknown, that are novel, that are through serendipity, often you're traveling a road that isn't well traveled. And many people do not want to be on that road with you because you're going off into a peripheral area that is very uncomfortable for a lot of people. For me and for many of my colleagues that now have come on board with what we have been doing, even Joe, we are more comfortable actually being the innovators, being the discoverers, being out there on the periphery of a field. We do not have to be in the center of a particular research field to feel that we have a security about what it is that we're doing. And at first, what happens is that it's a lonely road, but as you go down that road and other people start to pay attention to you and what your work is, they start to become interested. So all of a sudden, now you're gathering people about you as well. It's like a grassroots effort of making a difference.
So you had mentioned the word novel, and given current events and current climate of a lot of the things that we've heard of, especially over the last 3 years with coronavirus, could you give us a little more of a better definition? I mean, I know what you're talking about, but not everyone else knows what you're talking about when it comes to that word novel.
Okay, novel in the aspects of which I'm talking about really are the approaches that others aren't necessarily taking, so that you're now utilizing your scientific acumen or your scientific training to actually now pursue information that others may not be pursuing. When it comes to the issue with the coronavirus, which is interesting, is that early on, most people weren't really pursuing avenues of understanding of what coronaviruses are doing. In the early 2000s, there was the original SARS-CoV-1, which came out of China, went into Canada, hit into the United States a little bit, and across the globe. It was, again, a severe respiratory illness that was occurring. It did not become a pandemic like our COVID-2 pandemic, COVID-19, that we've experienced for the last 3 years and continue to experience, by the way. What's interesting here, though, is as these last 3 years have now come along and come to pass, people have been studying this process more avidly and realizing that it's not just a respiratory virus that is actually now that we're dealing with. It is a virus that is affecting us and infecting us through respiratory pathways, but it actually is leading to systemic disease, meaning it is actually going throughout our bodies. And in some ways, that's a novel understanding of how this type of infectious agent can actually affect us. One of my interests is, because I study Alzheimer's disease, is how is this virus affecting the nervous system? And unfortunately, we're finding that it is affecting the nervous system. And about 2 months ago, there was an article that was published, not by us, but by others studying this issue, that demonstrated there's an increased risk for individuals over the age of 65 that contract this particular virus to now going on to getting or being diagnosed with Alzheimer's disease within a year after the infection. So it's a really serious issue here that's being now discovered and uncovered. And now we're still trying to understand, how is it actually leading to that process? Well, my own work on infection and Alzheimer's disease actually really parallels what others are doing now with COVID and Alzheimer's disease, in the sense that in the late 1990s, we discovered in my laboratory and reported for the very first time that a respiratory chlamydia organism that we become infected with by breathing it in through respiratory droplets can actually now affect the brain. And I say it parallels the COVID issue because a couple of things occur here. One is that the respiratory chlamydia issue, which is not the same as your sexually transmitted chlamydia issue. This is a respiratory chlamydia that really just infects the respiratory tract. It can actually be inhaled into our noses and infect our olfactory cells in our noses. Those are the cells that are processing, the initial processing of your sense of smell. And COVID also, COVID-2 for COVID-19, also can affect those particular cells in our noses, the ones for processing smell. What's intriguing is that those cells in our nose for smell now go into the brain, or have connections that go into the brain to the what we call the olfactory bulbs, and then regions of processing of olfaction deep into our brain tissues. Those regions are the very first regions that are affected in Alzheimer's disease. So we believe that if you impact your sense of smell through certain types of infections, you could actually create the scenario of now damaging the regions in your brain that are earliest damaged in Alzheimer's disease. And those would be the hippocampus and your entorhinal cortex.
Interesting. Just because it's topical, one of our members in the chat here, Aru Warrior, said that he completely lost his sense of smell for a few weeks after he first got COVID. So that kind of goes into very similar to what you were just discussing.
Yes, and the loss of sense of smell, while it's disturbing, is something that can be recovered depending on the extent of damage there. It does not always lead to damage in the brain, so it should not be regarded that it's going to always lead to now your significant damaging insult in brain tissue. It can, however, lead to that. And this is what we're trying to figure out is why in some people could the individual lose their sense of smell and then regain it and not have any neurocognitive change. Then why in others could you lose a sense of smell and go now down a pathway and have neurocognitive change because there's something happening in the brain as well after being infected this way. And we're trying to figure out now, what are those other factors? Because we know this is not working in a vacuum. We know that there are other risk factors here. Some of those risk factors could be other environmental agents. It could also be a genetic predisposition for developing some type of change in in one's brain.
Right. Actually, this is very interesting and topical to what we're talking about. That's why I'm bringing it up. He continues on that after he got his vaccination, he regained a couple of smells, and it actually took him several months to get used to smelling those smells again. If I'm not mistaken, that's because the brain is kind of being reintroduced to a certain level of sensitization that it's no longer used to anymore. Is that correct?
That's partly it, yes, so that reutilization and regaining. But also, which is interesting, is in our olfactory neuroepithelial cells of our noses, those cells will degenerate and regenerate approximately every 90 days. On average. So it's possible that cells that were damaged due to an infectious agent could actually undergo a degenerative process, but with the regenerative process, where new cells are now taking the place, like stem cells now growing in and now taking the place of the old cells, those now have the ability to now pick up a sense of smell again because they're staying connected into the olfactory regions of the brain. So it is possible that you could regain it because of that regenerative capacity.
Interesting. And just a reminder, if anyone came in late, when we're talking about the olfactory system, we're talking about the cells inside of your nose.
Yes.
So, what was it like for you as a researcher? I know what it was like for me because obviously it was my perspective, but what was it like for you to go through the entirety of a pandemic? Now, I know for me, whenever the term pandemic comes up, it's always in terms of history. It's always in terms of the past. I don't think I've ever seen the word pandemic being used outside of a historical context before 3 years ago.
Yes, there were hints that we would be having pandemics throughout history. Obviously, in 1918, we had the Spanish flu. That was a major pandemic. But we've actually had a number of different types of pandemics, like polio, for instance, was a pandemic. And unfortunately, we came up with a vaccine for that. So these infectious diseases can become pandemics. So up until COVID-19, we thought that the next big pandemic would probably be something like the bird flu. And the bird flu is still out there. And this is because the flu virus can be picked up by different types of animals. It's interesting because in China, where they have a large agricultural society, pigs and birds actually carry their own flu viruses. And sometimes those flu viruses can now bind together in what we call their genetic material can reassort. So genetic material from the pig virus and from the bird virus can now join together and become now like a new virus. Sometimes when that happens, you can get crossover of the flu strains into the human population. Mm-hmm. If that happens and you have unique recombinations of what we call this H and N, so H, like for instance H5N1 or H7N9, these types of reassortments and combinations of your hemagglutinins and neuraminidase molecules can lead to increased infection in us of a flu strain. If that happens, humans can now be affected with a very novel type of flu, which could become a pandemic because it's a respiratory virus. Fortunately, that has not happened yet. However, there are predictions that these are still in the offing because of the globalization of our world. We now have 8 billion people now on the planet. That is projected to go to about 10 to almost 11 billion by the end of this century. So we are now, in a way, with all the travel and the interconnected societies, and just through our air currents, by the way, tens of thousands, if not millions of viruses can actually travel through the air currents and be picked up across the globe. Because of that, we are always at risk that respiratory organisms can actually now affect the human population.
So one thing that I've tried to bring up at least a few times on my stream is that while most scientists always seem to be the bearers of the bad news and all doom and gloom, I kind of try and state to as many as I can that it's a certain objective, almost clinical perspective that we're trying to place on the facts so that our personal opinion doesn't come through on those facts. So we're not providing our conjecture on those facts. Would you agree with that kind of assessment?
I do agree with that. This is based on data and facts and information that we uncover over periods of time. And as we uncover these particular elements of science and nature, we actually can then start to make predictions. And this is where the predictions of other pandemics in the future may be coming about, based on what has been seen up to now and what could happen. We're hoping it doesn't happen. However, that's a hope, and we are often surprised with nature and biology. The positive here, though, is that by understanding these issues, we are able to, as a society, as a group of, and this is where science comes in big time, is rapidly come up with ways of correcting the problem or treating the problem, or in this case, vaccinating against the problem. Look at how rapidly the mRNA vaccines came about that are effective and safe, by the way, in the human population against the coronavirus. Mm-hmm. Well, those mRNA vaccines basically were in development, not for coronavirus, but for cancer, actually, going back at least 20 to 25 years ago. And now they're being applied to infectious diseases and to vaccine development, and they're also being moved forward and will be applied toward cancers as well, because we've gained more information with the COVID issue of how the mRNA vaccines can be made, and very specifically made. And what's happening there is that, in this case, an mRNA is injected into your body, and what it's doing is it's actually coding for a specific region of the virus, so part of the spike protein of the virus, so that our body then can, as the spike protein is made in our bodies respond to it because it's foreign, and now you get antibodies made to it those antibodies are what's protecting us. Well, just think, in cancers, certain cancers express abnormal types of proteins, okay? Or if they don't express it, if you can express something that is novel in a cancer cell, our bodies will respond to it. So potentially you can actually use an mRNA approach to create a novel protein in a cancer cell to allow our immune system to actually attack the cancer cell, which would be great because then you can eliminate the cancer more of a natural way than having to use chemotherapy and radiation, etc.
So basically, if I'm understanding your statement correctly, you're basically saying that we were using techniques that were already in existence to combat something that was still very new. So it's almost like you had the solution before you had the problem, in a way.
Yes, and the solution was being worked on before the problem. And it's basically now taking the different silos and mixing the silos is exactly what we've done with Alzheimer's disease now, too, with our own research. Mixing microbiology with neuroscience and neurobiology and putting them together to come up with ideas here and findings that are going to help us solve the problem. And this is important for science now, is to take your silos and to combine your silos, to actually now use information interchangeably to actually make a difference. And this is where now science is actually really starting to focus this way, which is a really good thing, because this is how we're going to make changes and differences and discoveries and cures and treatments going forward for a variety of different types of diseases.
I know that, especially because my I don't know if you ever knew this, but my father was a high school history teacher for about 20 years, 25 years.
You had mentioned that to me, Joe.
But one of the things that he always told me was that history is a very cyclical thing in terms of the long game. So if you were to look at something over a much longer period of time, you would start to see these cycles occur. And one of the things that I've always noticed whenever I look back in scientific history is that you had naturalism, which was kind of the precursor to what biology was. And everyone tried their best to kind of be the generalist of everything, almost the jack of all trades. And then it seemed like as we were getting to the modern era, we started as a group, as a community, if you will, to branch out and to become the most intelligent person in one or two topics. And now it almost seems like that we're reversing course to that, and we're trying to come back together as a group to start saying, we found out this. We should really be talking about this together.
That's a really good point. Part of the reason for that, too, is that as scientists, in order for us to be able to wrap our brains about a problem, we reduce it. We reduce the problem to something that is understandable for us as individuals. And then we branch back out. My approach has always been to keep the 30,000-foot level in mind as I've reduced the problem to try to understand specifics of an entity. And then the 30,000-foot level, though, is how does it really apply with everything else that we know? So it's sort of like taking all these individual facts that are coming from different laboratories now and putting them together. That's why many of us believe bioinformatics is so important in today's world, but also using artificial intelligence and supercomputing to actually understand how facts that seem to be disparate from one laboratory to another could actually be combined together and now give us answers that make much better sense than ever before, because now you're taking all the information and curating it in a way that you now have a three-dimensionality of your information, so you can see it now how it's all interacting together.
Okay. So I know for me, certain techniques and certain equipment today is way more available than it was when I was an early freshman back in 2006, 2007. How has and trust me, I know that with the evolution of knowledge, it has changed. But how has science changed for you from when you were that bright-eyed young man walking into, your first class at university to now, where you're at in your career?
Well, what's interesting is that my career really goes back to if I started with college into the 70s. And in the 70s, discoveries were still being made about genetics and how different types of genes are being expressed, of how you could start to manipulate cells genetically. But it was very, very basic and very fundamental. Those fundamental aspects, though, have led over many periods of time and many years into a more sophisticated understanding of how you could actually now understand biology. So when I was in school, I was very interested in trying to see what I couldn't see, this microbial world. So I got interested in electron microscopy, where you can actually go down to more of a molecular ultrastructural level of seeing inside of cells and seeing the things that are around us or in us through a very special microscopy technique. But as time went on, I realized that, okay, we need to be able to now understand biology at all different levels. Not only at the ultrastructural level, you have to bring it back up to now more of the gross level. The body level, the cellular level, tissue level, body level, so that you see how everything fits together and how that works. And in the process, from the 70s until current, we have had so many valuable, remarkable discoveries of how to actually now understand biology. And I'll just use one example. Polymerase chain reaction, what we call PCR. Kary Mullis came along with this. He was a surfer out in California, but somebody who was really a great thinker. And he came up with PCR. And PCR is a way of understanding genetics, of actually being able to manipulate genetics so we can have a better understanding of what genes are. Once we know what genes are, we can then understand what they do and how they code for proteins and how the proteins work. Well, that was a revolution that actually led to the ability of us doing the Human Genome Project in the late 1990s, early 2000s, to understand what is the human genome, what are the other genomes of all these other organisms as well, and being able to actually now figure out what comprises this genome, which is, for us, it's a 3 billion base paired genome. It's huge. So in today's world, we can now sequence an individual's genome in just a couple of days and for a very cheap price. Back then, it used supercomputers in the early 2000s, supercomputers and running machines day and night for over years in order to figure out what the genome was. So these are just examples of how we've come from where we were and where we are now, and how the remarkable discoveries in the future are going to be made as well, because everything builds upon itself. We all, like the old saying goes, all of us stand on the shoulders of giants, and we just get taller because we keep standing on those shoulders, okay? And hopefully we can become giants of our own, and others will stand on our shoulders as well for the future.
No, absolutely. And, I can attest to some of the advances since polymerase chain reaction, because that's what I specialized on when I was a undergraduate, was doing a lot of polymerase chain reaction via a DNA sequencing technique that was referred to as D.D. Sanger sequencing. And I remember professors of mine telling me that this was going to be the technique that I was going to make my career on, and no one would ever I would never need to worry about getting a job. And then maybe 3 weeks after he had told me that, a completely different form of sequencing had came out in the form of pyrosequencing, which is a much more elevated form of what's termed as shotgun sequencing. So to touch back to a piece that we were just discussing before, Aru Warrior again says, so more or less we have way more information than our brains can realistically process. So we need to use tools to kind of get back around to condensing all the data into something that we can comprehend.
Yes.
Yeah.
Yes, we do.
Yes.
And that is a big push. And I think as as quantum computing comes into being, which I envision will be in the near future, realistically, where 10 to the 18th per second of information can be evaluated. We're talking about changing the whole understanding of so many different fields, not only biology, but any fields that you have, a lot of information that's been generated of how you can use that information. Mm-hmm. So I think that's going to help us putting things together.
Yeah.
And really, it's coming down to putting things together and always considering how do things fit. As new discoveries are made, how do they fit with the old discoveries? Which to me is very intriguing. By the way, some time ago we mentioned pandemics, and one of the ones I didn't mention, I mentioned polio, the Spanish flu. How about HIV and AIDS? That's a pandemic that's still ongoing, actually, for the last 40 years.
I remember being a high school student and them being afraid to call it a pandemic. Still, in early 2000s, they were afraid still to call it a pandemic.
Which is amazing, because it's exactly what it is.
Right.
And we still don't have a vaccine that's effective for HIV. Here you have an organism, another virus. It's a different type of virus, a retrovirus, that all of a sudden is creating havoc. Fortunately, over the years, there have been a multitude of drugs developed that can actually now treat HIV, so that now it is actually a manageable chronic issue. So if someone's infected, they can be treated so that they don't go on to develop AIDS, which is the acquired immune deficiency syndrome, and which would then lead to demise over time. So fortunately now it's a manageable syndrome. However, a symptom and disease. However, we're still working. People are still working on developing a vaccine that would be effective for HIV. But there are some viruses like this, and hopefully, we're getting a handle on this as we go into time. But some of these are very refractory or not capable of being manipulated, even like what we've done with the COVID and COVID-2 with COVID-19, to actually develop something that's effective in the human population as far as vaccine goes. But the knowledge we've gained with COVID-2 and COVID is now being applied to HIV, and hopefully a vaccine will come about in the near future.
So I know one misconception that I deal with as a scientist whenever I speak to someone who's never really met a scientist in person or in the flesh, so to speak. How do you phrase like your typical work day? So for me, it's I always get the question, well, what do you guys do all day? And one of the jokes I like to make is, well, we sit there in our chairs with our lab coats on, steepling our fingers, maniacally laughing. But how would you characterize what we do exactly?
So what it is we do is that we consistently work on problems. I would say that scientists are problem solvers, is the way to approach this. And problem solving has many ingredients to it. Some of the ingredients are just really mundane things of you're trying to design an experiment. You're trying to now see, okay, how would things work? So you sort of have a conjecture in your mind of how something could work. And then you go about thinking, well, how am I going to design an experiment of actually testing that? So it's sort of like coming up with a hypothesis, and now you're saying, well, let me design something to actually test it. Okay, so that's something that does occur. It doesn't occur on a daily basis, because once you've come up with one of these conjectures and hypothesis, you want to go to the laboratory and say, okay, well, what can I do with it? Okay, and usually you go down that path, and you say, okay, well, this might take some time. I have to order supplies. I have to make sure I have machines on board that I can use. I make sure I have enough people that I can also have work on this. So you aren't necessarily only in the laboratory yourself, you're there with other people. Sometimes we're not even there as the principal investigator in the laboratory. We have our technical help, we have students, we have postdoctoral individuals, we have other collaborators that are working on it at the laboratory bench. And we might be in our offices working on now trying to find more information about what we think the hypothesis is and see if others have thought of this. So we might go on our computers and go into PubMed or Medline or Google searching, etc., to see what others have done. Because to have a novel thought is really a novel, talk about novel, is a unique issue. Most thought is not novel. It's now combining different things together possibly that makes it novel, but most thought in and of itself is not novel, and you find that out by doing your different searches and talking to people, etc.
Are you saying that we're not just staring intently at beakers full of green fluid?
No, we're not.
I can't take credit for that one. Again, that's our lovely chat. No, every once in a while, you do get to do that kind of thing in that you see in TV where you freehand pour some liquid into something.
Well, what's interesting too is sometimes you do have a eureka moment, and I've had a couple of those in my career, which is really phenomenal, because if you have a eureka moment, you feel like you're like on the top of the mountain, that you've climbed Everest and you're right there planting the flag that, oh, this is eureka, mm-hmm. This is going to be it, and then you realize that you have to take a step back and go, okay, but that's just the first step of a eureka moment. And I can give you an example, is that when we discovered this respiratory chlamydial organism, when we discovered this in Alzheimer's disease, we actually found it in brain tissues from individuals who had died from Alzheimer's disease, and now we were analyzing their tissues. And we found it by electron microscopy. Meaning, we had to take the tissues and process them in a way that you could put them under an electron microscope, which is a real involved endeavor.
Yeah, that's a lot of hoops you have to jump through.
And then you just have a 1 millimeter square piece of tissue that you're looking at under an electron microscope. And you can imagine how many of those little pieces you would have to look at to find something. Well, this was a finding, actually, that a graduate student of mine had when I said, okay, sit at the electron microscope and call me if you see anything. Well, he called me, and what we saw was this infectious agent, because I was trained classically in cell biology at the ultrastructural level. Mm-hmm. I can tell you what things are inside of a cell if you looked at it under electron microscopy. And I said, wow, this is not normal. This is something that's unique. You found what we think is the chlamydial organism, the respiratory chlamydial organism. And by other techniques, we actually proved, using molecular biology, that the genes for that organism were of that particular organism in this brain tissues. And that's how we made that discovery. Well, that was a key eureka moment of, oh my God, we actually saw this organism after thinking that it could be there.
Mm. So another question that we have here, which again, very, very topical. So eureka moments are less, I've climbed the ladder, and more, I have found the ladder. Trust me.
Yes, well, that's a good point.
Trust me, you don't know how right you are. It gets even worse when you think you found the ladder, but you're at the top of somebody else's ladder who did all of that months or even years ago, and you find out that they've been proven right several times over by someone else.
And that's exactly what happens with science. So when we first published our work, we were the very first lab to publish on this finding, which is also a eureka moment, because that doesn't happen very often.
Yeah.
Exactly what you said, Joe, that others also are doing things that you may not know about. Mm-hmm. However, this was documented as being the very first documentation of this finding.
Wow.
Which is significant.
Yeah.
So we sort of planted the flag on that. Now, others coming along and finding it again is also helpful for us, because that goes to the replicability of science. That if you find something, somebody else should be able to find it as well, even though it might be difficult. And that's where others come along onto the scene, and some people want to, do the fast and dirty experiment to think, oh yeah, if I do it this way, I could find it too, and they may not. You may have to delve into the problem a lot deeper or experimentally, more rigorously, in order to also replicate what's been found. But over the years, what we have found has been found by others as well, and is now, we're studying it, a few other labs are studying it, etc.
Great. I know at least from my past, there have been several instances where I have kind of done an experiment, or I've worked with someone, and there's that flash in their eyes in that moment. And especially when it's someone who's young in their PhD, I will almost grab them by the shoulders and say, hold on to this moment, because this is one of those moments, that moment of wonder that kind of keeps you going throughout the entire process. At least for me, I know that I have like maybe 3 or 4 that I kind of hold on to with all of my might.
Yes. Yes, and you have to do that. Those moments don't come along that often. It's more of a drudgery, I would say, of doing experiment after experiment, and you find data, you generate data, you find results, but it's not as if it's something that is remarkable, or it has been something that is coming at you out of the blue that you never even thought about. Those things can happen, but it's rare. But the other thing is, you need to be observant. Even if you find something that seems out of place, that it was not a prediction of your experiment, you need to be able to understand that that wasn't a prediction, but could it be important? And a lot of people often will stay in the middle of the road and not delve off to the side, thinking that that could be important. And that's where discovery often can be missed if you don't take advantage of those observations that are made.
Right. I know for me, I've always kind of described, especially for my wife, because my wife is an artist, and it's not that she doesn't understand what I do, it's that it's hard for her to understand kind of what I do versus what she thinks I does. So, and my mother is absolutely the exact same way, because it's the what movies and TV has kind of made us as scientists look like. I mean, I love the joke whenever I watch a crime procedural with my mother that involves any kind of CSI, and 2 minutes after they get like a shoe, right, they come back with, we know exactly who this guy is, we ran him through the DNA database, and we've sequenced his DNA, and we know it's him 100%. And it's like, it doesn't work like that. It takes so much time. I've always thought of science as a collection of baby steps where it is a different day, but all the same thing, almost.
It is. And that's more accurate, really, to think of it that way. The baby steps. There are times where, as information is generated, that you want to leap forward from the baby steps and take giant steps. It's very difficult to do that, though, because you need to have a lot of controls in place. You need to have a replication of information. You need to have funding sources. You need to have the ability to continue on making those baby steps before the giant step is made. Now, those giant steps can take place. Typically, though, they need a group of people, consortium of people that have like-mindedness, findings that can help and support one another, and you then need to do things more as a group than as individuals.
One thing that I've started to see a little bit more and more, and maybe it's because it's one of those conversations that we're having more in the forefront as a society, but I've noticed, at least for myself, that mental health especially in scientists and researchers, is starting to become more of a I don't want to say a worried about thing, because that's not the right way of saying it, but it's becoming more of a looked upon thing, a thing that we are trying to treat actively, because I've seen some researchers who have one bad, review from one reviewer, and they go into a complete tailspin.
Yeah, this can happen. Part of the reason behind that is that science in just about every country, including our own, while it's being supported, it's not supported to the level that it needs to be. And a lot of people's careers depend on obtaining support that way. And I mean, support is dollars. You need to have money actually to do research. The equipment costs a lot of money. The supplies cost a lot of money. You need to have a salary in doing your research. So it all goes together. Most of the scientists in the U.S., for instance, try to get funding through the National Institutes of Health. Well, here's a perspective. The National Institutes of Health will fund about 20,000 grants on a yearly basis. Well, that seems great. However, the number of grants that are being submitted to the National Institutes of Health is about 60 to 80,000. So maybe 1/4, and I say maybe, because sometimes it is much less, even maybe 10%, of the grants submitted to the NIH are actually being funded. So you can imagine all the scientists out there doing their science, believing in their science, doing really good science, that aren't getting the financial support to move their science forward, or possibly even moving their career forward. So the stress levels are enormous. Mm-hmm. People are working under that gun of trying to now have the proper data findings written up into a great grant submission to get it funded through a governmental agency that has really failed, I think, over time, the scientific community. Mm-hmm. And I say this because of the way grants are reviewed and the way they're awarded. And it's limiting for a large percentage of the scientists out here doing science. So, what do scientists do? Scientists then will go off and try to get philanthropy money. They will try to work with biotechs, big pharma, come up with their own personal money, do crowdsource funding, etc., etc. Mm-hmm. All different ways to try to now keep going with their ideas. And I'm really sensitive to this issue because when we have had our novel findings of infection and Alzheimer's disease, the NIH did not want to fund us. We were funded one time through the NIH for a 4-year grant, and that's it. Over the last, I've been doing infection and Alzheimer's disease for the last 25 years. One time. And they would not refund us, even though we had data and we had publications on the work. So it comes down to other scientists are falling into this category as well. Mm-hmm. And it's really problematic. The NIH has tried to fix this for many, many, many years, and it's still problematic. We're still trying to find better ways to fund a lot of science that's being done in this day and age.
Yeah, that is always one of the difficult aspects. I mean, one of the concepts that's kind of starting to go out of fashion, and I'm very grateful for this, because it's starting to happen at certain levels of... that's a very good one. Give me 1 second, and I will ask that question. The concept of publish or perish is starting to lose its lifespan. I've started to see certain higher institutions of learning that are almost doing away with it entirely, simply because they're burning out almost talented young researchers at a very fast rate.
Yes, in fact, the percentages of people that are doing their postdocs, where it's like that mm-hmm. internship after their doctorate, of going on into an academic position, has been decreasing for years and years. It may be at about the 20% of those individuals now going into an academic position where you would need to go for funding through the NIH. The other 80%, they're going out into industry for the most part. They're going to biotechs, big pharma. Some are leaving science altogether.
Yeah.
They're going into scientific writing, or they're medical education writing, or other aspects of what you could do as a scientist, but it's not really doing science.
I had a classmate who was interested in graphic design as a hobby, and now they edit figures for textbooks. Simply because they have a background in this. So I had mentioned that someone had asked a question, and it was very on topic. Have you ever been, I haven't, thankfully, but have you ever been in a situation where you've been pressured to research or reach certain conclusions by a patron or a funder?
To reach certain conclusions?
Yes.
No, I've always kept my science where whatever we find is what we publish on, whatever those findings are. So regardless of how it's funded, it comes down to maintaining a level of integrity of the scientific process that cannot be compromised. That goes to another issue, though, that I was going to mention too, is that scientific fraud has become a big issue in this day and age. And part of it is for this reason, is that these large laboratories that have a large amount of money that they've gotten from the NIH often are funding large laboratories. They're pitting other junior investigators against one another to come up with data that fits their hypothesis or ideas.
Cutthroat.
And that cutthroatness has led to fraudulent science. And this goes to even Nobel Prize winners having to have papers retracted because there are either errors or absolute manipulations of data that are put into certain papers. And now we're using more and more sophisticated techniques to find out how these images have been manipulated or how the data has been manipulated, and it's actually leading to fraud. And to me, that's the worst thing that can happen in science.
Agreed.
And that's because nature and biology has a purity to it. We are here to understand that. We are not here to manipulate it in a way that suits our purpose. We're here to use information and to gather information and understand it from a point of view of what is nature, what is biology, what is it about us, what are these aspects. As we get more sophisticated with understanding it, then we can develop techniques of studying it. That's what all these new techniques are coming about to do. Okay, those aren't really manipulating the information. Those are now taking the information and finding a way of applying it.
Yeah, that's. I had a classmate who unfortunately did get sucked into a situation that you were describing, and became so desperate that they did end up fabricating something, and it's very disgusting for me as a scientist because, like yourself, I put so A.R. Warrior, you kind of struck a nerve for both of us in a way because we put a lot of effort, a lot of time, a lot of sweat, blood, and tears of ourselves. Sometimes it's even at the cost of our own personal lives. And there are people who are doing the exact same thing, but they're also fabricating a result in some way, shape, or form just to kind of reach that finish line just a little bit sooner.
Yeah, and often they're either found out or will be found out at some point in time.
The most recent article.
Yes, that's true.
There was an article recently where it was a figure, I believe, that tipped them off.
A few different figures.
Yeah, there were a few different figures where they just seemed just a little too good, if you will. And that's boiling down probably like an entire page's worth of analysis of these figures to a single sentence. The figures just seemed a little too good for their results.
Yeah, and this was an Alzheimer paper, actually, which is a shame because a lot of information was based on that information. So that as people went on experimenting, they were using that previous information to help guide them. Fabrication to me, science is not a fictional process. Science should be nonfiction.
It's a factual process.
Right. It is one where it's like writing about history and using information to write about the history that occurred. So it is factual. It's something that did occur. It's there. It's real. You can touch it, you can feel it, you can sense it, you can see it, etc.
Yeah.
And that's what we should be all striving to do, is to understand biology and nature with what we do, not create something of our own mind as if that's going to be now biology and nature.
Right, and that brings me to one thing that I've almost lit. So, as you know, because that's kind of the role that I fill in our kind of small little sphere here, I focus on data so much down to almost a fault, because for me the data is the data is the data is the data. It is one of those immutable constructs of scientific research that once the data is there, it's there. It's like a human being or a being in and of itself.
Right.
Once it's there, it is there, and you almost need to advocate for it, advocate against it at times. You almost treat it like it is a living being in a way.
It's so true, because that becomes now a framework. That becomes the information that you're going to use to hopefully explain or further understand what it is you're studying.
Yeah.
And so now that it's sort of like now you're collecting more and more data to help you with the better understanding. Okay, and you're not saying, oh, let me make up something for better understanding. It's actually, okay, let me study something for better understanding. Let me do it in a pure way so that you're really now getting the essence of what nature is. That's what we have to understand. Because we could all go off and write a fictional account of what like the Andromeda strain. Let's come up with the Andromeda strain and say, oh yeah, this could it could
Very
become
topical.
reality.
Very topical.
Right. It could become something more like reality. However, that was fiction. Okay.
Yes.
But
it's
it was
fiction that got a lot right, which is very interesting.
That's true. But it was using information, but it was also now fabricating things along the way to sort of like manipulate that information.
Correct.
Versus just an understanding, as we do as scientists, to understand what is it that we have in front of us. Let's figure it out. Let's problem solve it. Right?
Yeah, no, absolutely. And I mean, that's kind of the interesting love-hate relationship that I kind of have with science fiction and fantasy in general, because there is so much that it gets right, and then you'll sit there and you'll almost be like, yes, now get the next thing right, and they don't, and it almost breaks my heart when it happens, because it's like you were so close to what it's actually like. So one of the things that I wanted to get your opinion on, one of my longtime followers of the channel and subscriber for a period of time, his handle is Alfarius, which is related to something completely different, but his name is Casper. Great guy. He specifically asks me, he's got this lofty goal, almost rooted in science fiction, and I think it's actually, in truth, it's a great idea. It's almost, what he's attempting to do is he's attempting to almost theorize a methodology of creating an augmented limb for someone who is missing a limb that can feel and provide feedback from the limb's, I guess, sensor, if you will, but in a way that the entire limb is very analogous to the human arm, where the entire thing is able to sense, not just a few touch pads or pressure sensors on the fingers. How much science fiction do you think that is for us, and how much science fact do you think is actually rooted in that?
Well, I think that eventually will be a reality. And I think that because even of today, artificial limbs have been able to be attached into the nervous system, our nervous system, the human nervous system, and have a level of functionality. Our nervous system also, for individuals that are paralyzed, can't speak, you can actually put electrodes into the brain, different areas of the brain, to actually elicit a now thought and speech from individuals that now are debilitated that way. So there are a lot of different ways that the brain is being now looked at, and our nervous system in general is being looked at and explored. In addition to now having robotics and humanoid-like limbs, features about our bodies, etc. So while we know through science fiction that they've come up with these types of things, I actually think this is sort of leading us along the way. We will get there at one day where we will be able to use this more practically speaking, with connection to our own nervous system.
Interesting. You know, one concern that I have, and maybe you can tell me if I'm way off base, is the level of sensory overload that something like that would potentially provide to the person, at least in the very beginning. Just the amount of information that is coming through. I almost wonder if it'll blow the circuit of the actual limb itself, at least in its early days. I know that especially when we have oversensitization, the brain has almost a deadening function where it almost silences certain pathways to say, okay, we need to protect ourselves and insulate ourselves. But do you see something like that ever becoming a potential pitfall?
Well, I don't really know enough about it as far as how to modulate the input. But I think of like electricity and transformers and things of that nature, of how now you can actually now modulate electricity coming into a home or a business, whatever.
Yeah, it's becoming less of a mechanical source these days.
Right. So in some ways, I can imagine that type of an approach would be used so that you wouldn't have that type of overload situation. And then again, the brain is... So this is where the brain actually can have a level of plasticity to it, or ability to take in information or be utilized in a way that it can now evolve. It can adapt to how cells are actually being triggered through chemical and electrical reactions. And depending on how you do that, the brain will adapt to that environment. It's sort of like in epilepsy, for instance. If you have now ictal types of activity where it's like just single cells now firing indiscriminately, leading to now an output and then someone having an epileptic type of response. How do you modulate that? Well, you can modulate that through structure, by limiting then the synapses that you would have there. You could also modulate it by affecting the electrical activity, or you do it through surgery now. You take out the damaged cellular area, and now it eradicates the problem.
So one thing that was just brought up was a warrior says, I saw something that solved color blindness was cured or worked around by using a camera and electrodes to deliberately give someone synesthesia.
Oh, that's
I've seen something very similar. It almost looks like if you've ever seen Microsoft Glass or one of the early wearable PCs where it was almost like a camera that would sit here, and then it would force the information through certain electrodes. You had to wear a controller on your hip, but it allowed someone to experience color similar to how we do. We're getting into aspects of the brain that I don't know if you're completely, 100% sound on. Not to say that you're limited in that regard, but
well, I don't study synesthesia, but it's interesting, and the wiring in the brain is phenomenal. And now we have hard wiring, and then we have some levels of flexibility and plasticity. So the hard wiring is like our optic nerves and our acoustic nerves, all of the cranial nerves. They're all hardwired. However, in development, that wiring can actually not be connected properly. And so you could literally hear color or see sound. And why is that? Because of the wiring of where the connectivities were being actually made in areas of the brain where they should not have been made, actually.
Well, if I'm not mistaken, there's actually I believe it's a form of science that is devoted to, if you've ever seen where they put the sand on like a piece of glass or something, someone takes like a bow or something and you get the pattern.
Yes, you get the pattern.
Yeah.
Yes.
I guess that's actually seeing the waveform, if you will, in a more in a way 2-dimensional, but also
That's
in a way
an interesting,
3-dimensional
way of looking at it. Yeah. It's an analogous way. Yeah.
So another question that he had was going back to something that we had discussed earlier. And yes, I am watching the time for you, so don't worry. Dr. Balin has a lecture that he's got to give this evening. So to get back to what we were talking about, the genome sequencing, have you ever encountered in a situation in your research where there's been any correlation between identical twins and Alzheimer's with their genomes being so similar? Now, I can offer certain aspects to this in that identical twins in terms of genome is kind of the genetic holy grail because you want to find those individuals, and unfortunately, if you did, they would instantly become lab rats for some geneticist somewhere.
Yes.
Unfortunately. Because there's just so much, at least for me, just the idea of a true genetic copy of something, it's, to use a buzzword, it's almost titillating in a way.
Yes, it is. With regard to Alzheimer's disease, well, there may be genetic risk that would be similar in identical twins. That's where you'd have to look at environment. And I'm trying to remember there were a few twin studies done. And it wasn't so much that the twins always went on to develop the same disease. And I believe from what I recount, is that if they had been separated and living in different environments, it was shown that one may develop disease and the other one may not. And it goes to how the environment and genes, or nature versus nurture, is actually influencing outcome.
Yeah, there's been a couple of anecdotal situations. I think there was a couple of twins, but I know that more recently there was triplets that were actually brought back together, and they were actually they were sent out of almost to 3 different, completely different forms of life. They all had glasses. They all had the same kind of tic that they did that was behavioral. It was very interesting to see from at least a genetic point of view, because there are certain aspects that just aren't coded into our genetics.
It's very true. Also, aspects of being identical is interesting, because does it mean that in each individual, every cell is going to express the same allele? In other words, because we have 2 alleles for each gene normally, one may be expressed when the other is not. So that's the typical. So
it's almost random too,
which one can get
expressed.
Right. So in identical twins, at least physically, they look identical. Now, do they think alike? At times you may find that, but maybe not complete. Also, what environment are they? If they're in the same environment, would they have the same risk of getting the same disease? It's potential, yes. Also, the potential is that not necessarily. So because, and again, it goes to how the cells actually deal with more of an epigenetic or an environmental influence. and how those individual cells in that particular individual, even though they're twins, actually could behave a little bit differently.
So, just for everyone else, could you further explain the term epigenetics?
Yes, so epigenetics is actually where there's an influence on our genes that is not, it's coming from like an outside source, or it's a modification of how a gene is particularly expressed. For instance, if you had a chemical modification, if somebody put a molecule on a gene, it may then either limit its expression or enhance its expression, etc. And our bodies, in our cells, there are chemicals that can do that, can bind onto our DNA and influence how it actually then works.
Mm-hmm.
You can also have things like radiation and chemotherapy and immunotherapy, other types of things that could actually influence how our genes work or don't work. Those are things that influence the DNA, though, and the what makes our cells work the way they do. In fact, putting in the mRNA vaccine actually now is putting in something extra. It's not epigenetic, though. It's actually now utilizing our genetic machinery to actually now code for a small molecule that could help us by being presented to our immune system, and our immune system developing now antibodies to it, etc., to keep us healthy.
Yeah.
Okay, so there are different ways that our genes can actually be influenced. There's also another molecule called microRNA. And microRNA is something that's come onto the scene in about the last 10 to 15 years. It's a type of genetic material in all of our cells that can also influence how our genes work. And some of those microRNAs can actually keep us healthy. Some of those, depending on how they're expressed, could actually lead to disease, like a cancer, for instance. So the genetic aspects of what we are still being discovered and trying to be understood to a better degree.
So while I give our viewers, our chatters, a few more moments, if you could come up with a couple more questions if you'd like. We're going to be ending very soon. I want to give you a couple of moments. Is there any questions that have... well, God, I'm really going to put my foot in it this time. Is there any questions that you've ever had for me that you've wanted to ask that you never really have gotten to ask?
Well...
Other than my big mouth.
No, well, I guess I would like to ask you of where do you see yourself going here in science at this point in time? You're a really intelligent person. You can analyze data very well, and so that would be my biggest question.
Casper, I answered your question, buddy. You missed it too. He just popped in, unfortunately. So where I see myself, oh God, this is the one question that I always hate whenever I get it in an interview. I like to think of data and science almost as an ocean with tides and currents and waves, and I kind of go where it takes me. Almost I go with the flow of where the information takes me. So personally, I always let the data kind of, again, I like to think of the data as almost a entity in and of itself. I let the data kind of tell me where I need to go. I allow my brain to kind of extrapolate. And yes, if you haven't guessed, Dr. Balin has always been on the receiving end of several of those conversations. The brain soup making information. Yes, exactly. These are things that I tend to bring up to you, mostly because I'm still, the way that I was just having this conversation with someone else today, in terms of scientific age in my career, I'm still in diapers compared to a lot of the other researchers that still sit as scientists. There are still some, for lack of a better term, boomers that are sitting there still holding on with all of their might to their little chunk of the scientific world. And then there are some that are being very progressive that I've seen over the years and are actively trying to develop people like me that are younger, that are showing a interest, not just a interest, but an animated interest. And whenever I see that in our students, I try and nurture that as the best I can. So I kind of am getting the best of all the worlds that I want currently. I'm able to teach some of our students in my own little way. I'm able to answer the questions that I'm looking to answer, and it's really just where the questions are going to take me next.
Well, and that's a good approach to have.
Yeah.
I asked you that question too because you have, as a young person, you have projected many years ahead of you of ways of seeing the world, of how the world's going to change, and how you're going to now adapt with that
Right.
as well. As an old guy, I look at it that I can be a mentor, an advisor. We gain information from everyone around us, from our young people, our old people, and that's a good thing. And that's what I like to do. I like to hear what your ideas are. I like to, when we talk about things, because it helps in an overall understanding.
There's a great difference of perspective, and so the reason why I say that is because one of the questions that just came up was what advantages do you think you had growing as a scientist that younger scientists don't now, and to me, vice versa. I know that for me, at least with our conversations, it's the difference of perspective that you and I have. I come from a bioinformatic, genetic, and microbiological background. Also, I made my bones in inheritance. Yeah. That's what I focused on in graduate school because phylogeny, as it was termed, I think it's still termed as that now, was very big in the late 2010s. It's not so much anymore because we're not worrying about so much where we got things, but we're kind of worrying how they're evolving now. How has things changed so much that you kind of I don't want to say that you feel bad for the younger scientists, but how have things changed that you find it different for them?
I think it's much harder now than ever before to stay in academic science. Because academic science is one where you have responsibilities not only for your research but for your students and for your institution. You have to be really more well-rounded in dealing with the all of the issues that come up in academia. And the biggest thing I think too I'd go back to is the funding.
Yeah.
Without proper funding, without enough funding, without support, and doing it in a logical way of bringing along young scientists, without proper mentoring, you're left to sort of be hanging out there in the wind. And that's why I regard what I do also as a mentor. I feel that it's one of my responsibilities is to mentor younger people and help you as a guide. Now, I can't tell you what to do, and I would not tell you what to do. But it comes down to that I can help support how you would approach things, how you would go about doing things. I also help because now I sit on study sections now through the NIH and through the Infectious Disease Society of America, etc. Okay, so study sections are where we review grants to award money to people doing science. Well, I go out of my way to try to fund science, to try to see the positive in the work that's being done, not pick out negatives, because everything has a negative. Okay, no science that anybody is doing is perfect. It comes down to perfect design or whatever. It comes down to that: is there proper intent? Is there effort? Is there an intelligent approach, etc. In moving forward? And so that's what we like to pick out, and that's what we like to help support.
So, just to recap very quickly, because we're getting to 5:52, and I want to kind of get this. I don't want to say I want to wrap it up with 5 minutes to spare before your lecture.
Okay.
But I do have a question for you.
Okay.
About time and fate towards invention. What's your view on time and fate beginning in invention? So this kind of extends to a conversation that Casper and I had a while ago about limbs, because we were discussing things, and my concern was the limitations of the technology that we exist in.
Well, there are limitations in existing technology. However, I think technology is advancing a lot faster than we can even handle emotionally as humans. You know? That's the other part.
No,
so I
true.
think the technology is coming about to where we want to try things, and now it's the ability to do it. Is having the right population, experimental. We can't experiment with people. However, in some ways, as different inventions come along, especially with artificial limbs, artificial intelligence, of how we're using that in the human population, it is a human experimentation process. Okay, so in some ways we are doing that, but we can't. For instance, I can't put a drug into somebody experimentally without actually testing to see if it works and is safe, etc.
Exactly.
So in those cases, it's a little bit different. But to me, invention is happening daily, and it's a good thing. It's how we're applying the invention. How can we get it there to the population faster is the key. And that requires, having groups of people working on it, having funding sources, having people interested. And the funding sources can be of a variety of, it could be Elon Musk, for instance, funding some advanced technology to interface with a human brain.
Right.
Okay. Some of that is ongoing now.
Yes.
Okay, I don't know if through Musk or not, but it is ongoing now. So some of it's going to have to come through philanthropy, government, pharma, even in that kind of realm.
Well, if you could, as we're getting ready to send ourselves off, because it is 5:55, could you just quickly sum up for me and for those who might be joining us a little late, what it is that your research is currently that you're willing to share or able to share with us, and anything else that you'd like to say? I mean, the floor will be yours after that.
Okay, so the research that I do is on Alzheimer's disease and infection in Alzheimer's disease. And literally, we are looking at an organism, a chlamydia pneumoniae organism. It's a respiratory chlamydial organism, not the sexually transmitted form. I keep emphasizing that because when people hear chlamydia, they often think it's just sexually transmitted organisms
and
that disease. Right, that's trichomonas. This is chlamydia pneumoniae. Years ago, in the late 1990s, we first published on this, finding this organism in human brains, and those are the human brains were from people who had Alzheimer's disease. And we saw that it was not in the normal brains or the control brains that we evaluated. So we feel that this organism is an ingredient in Alzheimer's disease. Now, how is it an ingredient? Is it causative? Actually, I think it is in some cases. Now, that's a bold statement, but I think that we've been able to discover that this organism is an intracellular bacterium. So if you can envision a virus, this is a bacterium that's acting like a virus. It actually invades our cells. It comes into our bodies through respiration. It comes into the olfactory regions of our noses, sense of smell, can get into the brain. We found it in the entorhinal cortex and hippocampus and other areas of the Alzheimer brains we evaluated. We think this could actually create the scenario of generating the amyloid peptides that are often and usually found in Alzheimer's disease, and also the inflammation that is now occurring in Alzheimer's disease and is well accepted. We've been able to show that microglial cells actually can become infected. Microglia are the macrophages in our brains that actually control any infectious process that might happen in our brains. Those cells can actually be infected with chlamydia pneumoniae, and when they are, or they're exposed to the organism, they make the pro-inflammatory cytokines, the chemicals that actually damage nerve cells. we think that is the crux of the process that's happening with this particular organism. And we think it parallels what's happening with COVID-19 and CoV-2, even though the virus, CoV-2, does not get into the brain readily. It gets in a little bit, but it's generating inflammation in the human brain and can lead, by the studies that are already out there and are coming up, to be published in the future, can lead to now increasing the risk for developing Alzheimer's disease. So we think infectious agents actually can cause Alzheimer's disease and probably many other neurodegenerative processes. A lot of the frontotemporal dementias, maybe even some vascular dementia, and if this is the case, then this is revolutionary to our understanding of neurodegenerative processes that many of them, if not all, would have infectious components aligned with them. And that means we can find ways of treatment, of potentially prevention and treatment, and actually stopping these processes from happening in a large percentage of people across the globe.
Well, I want to thank you very much for joining us, Dr. Balin. We may have to do a part 2 of this at a later time.
I will be happy to.
One thing that you can probably guess just by seeing some of the interaction. Yes, so Casper, there are a few institutions that do put lecture series up on their websites. I think MIT has a couple of lecture series that are available for free, and I think it's their undergraduate programs. I don't know how extensive that is. He asked if it was possible to listen to a lecture from a distance. But I think we're going to have to do more of this. I think we'll need a part 2 for you, Dr. Balin.
As you could tell, I like speaking about the science in many different aspects of what it is we do and aspects of science. So I'll be happy to.
This is one of the things that I've been saying to the viewers of my channel, is that we as scientists are excited to talk about what it is that we do and what it is that we research. But anyway, again, I want to thank you so very much for joining us this afternoon. I know you have to get to a lecture soon.
Okay.
Very soon, actually, because it's 6 o'clock right now.
Okay. I'll be good.
But thank you very much, Dr. Balin. Well, I greatly appreciate it.
Thank you, Joe, for having me today, too.
It's my pleasure. It's my pleasure.
And thank you to the audience.
And thank you, all of you, for joining us. This will be available on our YouTube page. It will also be available for a limited time on the Twitch page as well. I want to thank you all for joining us. If you enjoyed this content and would like to see more of it, I hope you'll follow our channel. If you really enjoyed it, I hope you will subscribe so that you will be given better notifications, and also you won't have to worry about any kind of ads. Also, don't forget to join our Discord. We don't always just talk about science, but sometimes we will talk about video games, we'll talk about popular culture, and you can watch me be terrible at video games. So, thank you all so much. So long. Thanks for all the fish. Bye-bye, guys.